Yo, what's up? Explore privately. That's what you're gonna do with Tor, okay? Now, I'm not advertising Tor here. There's a lot of issues with Tor, okay? I'm not gonna get into all of them, but for what I have to do right now, it's like the best option. And actually, all I'm doing is just making a video. But, I don't like PII flowing all over the place by accident through my videos, and I get messages in the browser. I use Slack, other messaging platforms, so, yeah, this is fine. So yeah, I had an interruption in my video yesterday because pretty much the time of day which I record videos affects whether my video is going to actually go to completion, whether I'm going to finish what I'm talking about or not. So yeah, I had a little hiccup, but it's fine. I'm gonna get back on the ball. The entire last week I couldn't make videos, so those of you who are excited to see me speak and exposit the secrets of life, then listen to me, okay? Right now, and just keep watching, okay? It's gonna keep coming regularly. I got it. I'm working on it, okay? So, what am I gonna talk about today? I left off yesterday at Aquaporins, okay? So what is an aquaporin? So let's talk about a little bit about diffusion, okay? Actually, I left off at diffusion yesterday, not aquaporins, but we, still have to get through diffusion. Okay, so what's diffusion? Why is it important? So as I was saying yesterday, diffusion is a natural process where particles go from one area of high concentration naturally in the absence of all other forces to an area of lower concentration, okay? And again, don't allow. It just pretty much occurs naturally, right? And therefore it's a passive process which increases the entropy of the universe, right? And so there's a gradient established, a diffusion gradient established, and thus things will flow through. Another nice thing about diffusion is that diffusion of different objects occurs independently, typically. So if I have these these black objects here, and then I have these red objects here, right? Eventually, diffusion forces will occur independently for each object, right? Or will appear to occur independently for each object, such that there's a distribution of objects on both sides of this container that is equivalent, so that there's pretty much the distribution of items will be equal everywhere, and it'll be independent. So just because this species A is in here and species B is in here, their end result will be that their diffusion processes will not appear to interfere at the end of the day. Okay, the concentration of both will be equivalent on both sides. So that's really good to know, right? And that occurs in both directions. If B was here and going back this way, an equilibrium would be established such that the concentration on both sides of it would still be such. So if I put in a lot of A into one side and a lot of B into another side, you'll have eventually an even distribution of A and B on both sides. Okay, it doesn't matter what side you put the A and B into. Okay, now we're saying that this is a regular container. I drew this line just to create like a dividing line. I don't want to confuse you because there is a type of diffusion. Diffusion has two basic, explanations, or two basic forms that are explained to people typically. So the other form of diffusion is called osmosis, right? Okay, osmosis is specifically the diffusion of water. Okay. And often osmosis is talked about in the context of things like semi-permeable membrane. So you might have heard the term before, okay? Now, we'll use water here in this case, in this version of diffusion, but we don't need to use, technically this should be applicable for anything that's similar to water, or any other similar kind of structured experiment. So let's say we have a bunch of water molecules, and I'm just not going to draw the individual water molecules. I'm just going to draw water levels here. Okay. Let me see how much I'm recording so far. Okay, I'm recording. I'm not even sure. This screen flicks is just what it is. So basically, have a bunch of water here. What's a good way to draw the water? I'll just use this big spray paint tool. This is water. Oh, that's actually really cool because then the individual molecules you get a sense of the individual molecules, even though they're not. And then you have the water here. So typically, if this semi-permeability means that there is a permeability, which is the ability to pass through for certain types of substances, but not others. Semi-permeable. Allows passage of one type of molecule, but not another. Okay, that's the basic definition. So if this is a water permeable membrane, eventually the concentrations will get to the point where water is equal on both levels, both sides, right? So this will go up, this will go down. Okay. Now we want to repeat, undo. Okay, history. Okay. Oh wow, this paint program needs some work. Okay, so in the case of semi-permeability, right, what we want to talk about is multiple different things. So if we have water, let's say this is evenly divided. This doesn't look evenly divided, but let's just say it's evenly divided. And we have water level A, water level B, A, B. Now, what will happen if, let's say that they're equal, A and B are equal, okay? Now, what happens if I drop a bunch of molecules in A, molecules that are non-permeable to the semi-permeable membrane? So let's say I have these red circles in A. Okay. So what will happen? Now this is where it gets interesting because water from A, so if A and B have equal levels to start out with, and I drop this in A, these red balls in A, what will happen is water will actually flow from B to A, okay? And you'll see that the water level of A will rise and B will decrease. Okay, remember this is permeable, semi-permeable to water, so that means water can flow through each side, but the water can flow through each side, but these red objects can't flow to the other side. So this is really interesting. This is basically what's osmosis is called. Okay, so you can call these red molecules, we'll call them solid molecules for now. We have water diffuse from the right to the left, and therefore anything with the solid in it, any side with the solid in it, then where the solid is not able to distribute to the right. Remember, if this was fully permeable to the solid, then the solid will distribute to the right, and then the levels would still be equal on both sides of the container of A and B. But since the solid molecules, these red, spheres on A can't go here, we will have to this diffusion gradient will naturally, and this is again a natural process. It's not energy driven. It's not driven through input energy. It just will occur naturally to lower the energy of the system. Will occur such that more of the water gets, you can say, absorbed in the left-hand side because the water can flow through, even though the red molecules can't. So that's interesting because it's like now it looks different, right? That the water level is higher on the left side versus the right side. So the reason for this is actually quite complicated, okay? A lot of it depends on the solid itself, but you can imagine if you're looking at like a zoomed in view of what's going on with the water molecules. Let's try to create a permeable membrane here. Okay. I need to get some skill with these tools. Okay, I'm going to create a semi-permeable membrane. Okay. Semi-permeable is distinguished by these, gaps in the membrane. We create a semi-permeable membrane here for water, and then you can see I'm going to actually explicitly draw the water molecules, H2O, and I'll just call these some other molecule. So what happens is that, undo, oh God. So what happens is the water molecules will flow into the left-hand side, but these larger molecules there in simple terms, you can think that depending on what they are, they can trap the water molecules. For example, typically what people talk about in the situation of osmosis is this is some kind of positively charged sodium ion or something like that, right? So you can imagine that the water molecule, because of its partial charge, will get kind of attracted to the sodium, and in effect in water, a shell of water molecules forms around this sodium ion, right, or this positively charged ion, whatever it is. And this is called a solvation shell, because basically that's the basic principle of electromagnetics, right? Everything and physics, everything gets charged and it moves towards until this thing is crowded out and no more water can move towards it. So pretty much every positive ion gets trapped in a similar manner by water within itself. So this trapping of the water means that this water is not going to flow back. This trapping, electrostatic trapping, is much stronger, typically speaking, than the concentration gradient going back across the potential, going back across the permeable membrane. So water will not go back this way. It'll just get stuck. So you can imagine, right? The more solute here, the more water gets stuck to the solute, right? And the less it wants to stay on the left-hand side. So the level keeps decreasing and decreasing and decreasing, right? And that's it's a little bit of a jump, but that's why you can imagine that when they're talking about salting the earth, right? Like, say you throw a bunch of salt in the ground and plants start to die. They start to wilt. Why is that? Because the way plants work is that, the way specifically the water in plants work is that the water keeps the plant upright. The flow of water keeps the plant upright. The water's ability, the plant cell's ability to trap the water and make themselves rigid due to the incoming water is what keeps them straight, and that's called the turgor pressure. Now that happens because of osmosis within the plant cells, right? So the concentration of these, solutes happen to be in such a way that the plant cells can actually draw water by this same process into them, okay, and fill up and make themselves more rigid. What happens when you put salt, though, outside into the roots is that the roots, where the water gets drawn in from the ground, will no longer be able to just passively pull the water in, right? Because the plant cells are no longer, there's this now additional external force, which is due to the salt, the concentration of salt outside of the roots, right? Imagine, let's try another one. It discards itself anyway. So if you have a plant here, right, and you have these roots, right, and you have now I'm gonna put these salts here. Well, let's just call this ground. Okay, I'm gonna draw the salt layer. This is just ground, and you have water in the ground, right? This water is going to get drawn into these roots through, the mechanism is essentially, it's a little complicated why the roots draw the water in, but essentially the mechanism is that roots do it passively. The water passively goes in through the root cells and then goes in, water flows up the plant stem, and then the cell walls of the plant become rigid because of the water coming in, and then water gets pushed out again by a diffusive force through the stoma of the cell of the plant. But what will happen is, if I now put this gray colored salt in here, like this, if you put this gray colored salt in here, the ground is now going to be the side of the container, right, that has more solute in it, and the water is going to get trapped and intermingle with this salt. And in fact, besides being less available to the roots in general, it's actually going to pull, due to the hypertonicity of the soil, it's going to pull water out of the roots. And when water pulls out of the roots, the plant cells no longer have the ability to remain rigid. They're flaccid afterwards. And the flaccid plant cells then make the plant wilt, right? Because this is, the plant is no longer maintaining its turgor pressure, and therefore there's nothing strong enough to keep it straight. So basically, that's why salt, in a nutshell, will kill plants, right? Because at that point, none of the plant processes can go on. So that's kind of a way of describing osmosis, and that's the common way that people describe osmosis in a global sense. Okay, so let's go back to now that we kind of understand osmosis, let's go back to the cell walls, right? So the cell walls, right, cell membranes are phospholipid bilayers, and they have integral proteins in them that do things, okay? Most of these proteins are these transmembrane because they're going through the phospholipid bilayer. Most of these transmembrane proteins are responsible for the transport of different molecules into the cell, which can't normally be transported into the cell. Okay, the transmembrane protein would go through the entire membrane, not just one side. So, like, for instance, let's see what can go into a cell. So typically this phospholipid bilayer, everyone knows, is this phosphate head is a polar head. Polar head. These tails are nonpolar fat tails. Nonpolar diglycerides. Okay. And outside, again, facing the inside of the cell, are polar heads. Okay. So basically, polar heads means that any charged species, right, partial or otherwise, due to some charge balancing, is going to get attracted to these heads. And these charged species are not going to want to travel through naturally this place, right, this interior. It took me a while to really fully digest why that wouldn't occur. I mean, these are hydrophobic, right? These tails are hydrophobic, meaning they have no interaction with water. That's what hydrophobic. They're not repulsive. They just have no interaction with charged species. Okay, specifically water in this case, because that's the major solvent of these cells, right? So the thing is, like, one of these charged species, plus if these nonpolar heads was here through just like diffusion or whatnot, they would travel, right? These smaller charge ions, atoms, whatever, they would travel, but because they're being blocked by this polar head, they don't have any incentive. They're being electrostatically blocked by this polar head. They have no incentive to travel through the nonpolar region. They'll just aggregate up here, typically. Okay? Now, that applies for strongly charged species for sure, right? Because it's a while before they get to a place where their energy can be lowered, which is the nonpolar section inside the cell. So they'll just stick here. Now, this is true, right? For most, right? Like, remember, this is a continuum, okay? So what happens is, when the charge buildup is large enough inside, okay, then some may sneak through, right? Because you have a pressure potential causing these species to sneak through. But typically, that's not going to happen. That's why you have a special channels to allow these species conditionally to get through. Okay, now let's go back to what type of molecules. Let's not jump ahead too much. What type of molecules can go through the cell membrane? So I'm going to draw our cell membrane. And by the way, I'm drawing these cell membranes in a way that they're they're kind of rigid looking, right? But we should know that the cell membranes are actually part of a theoretical structure called the fluid mosaic model of the cell, meaning that there's a whole. It's actually, the cell membrane is not a solid. It is a floating, ever-changing mass of different proteins, phospholipids, structural lipids like cholesterols, integral proteins, other things. Okay, so it's always moving, but just to not confuse you, we'll just make it look static for now. Okay, so so water. Let's talk about water specifically. What we want to know: what kind of molecules can typically make it through the phospholipid bilayer? Okay, so small molecules can, like oxygen, molecular oxygen. I drew it really big here, but molecular oxygen is tiny. Okay, that's why, and it's good it's tiny because you need oxygen to breathe, and this is what this is all related to. Molecular oxygen can pretty much easily make it through because it's not charged. There's no charge and it's small. Okay, and if there's a larger concentration of oxygen outside of a cell, it will have a diffusion within to the cell, right? The same with carbon dioxide. Carbon dioxide is a small, very small, non-charged molecule, and therefore it won't be stuck at the hydrophilic part, which is the phosphate heads. It will just pass through along its merry way in both sides. Okay, and it doesn't care about what's happening in the middle of the cell membrane. It doesn't care what's happening outside. It's just going to go in and out as it pleases, or as physics and chemistry and biology demand. So what though will be stopped? Let's talk about water. Water is a small molecule. Okay, so normally water should go through, like based on the physical dimensions of water, which is tiny. Water should be able to get through because it's an oxygen and two tiny hydrogens, whereas 2 oxygens is larger. So it should technically get through. It should technically get through easier than oxygen, right? Alone, molecular oxygen. But it gets stuck here, right? As we were talking about. Now, water gets stuck for the most part, but water is what's known as partially charged. Okay, if you look at a water molecule, oxygen, hydrogen, right? And you'll often see in high school chemistry, you'll see the charge difference in water is partially negative towards the oxygen side and partially positive towards the hydrogen side. This means this dipole, right, has so many implications for water in its chemistry and its physics. But for the purposes of this video, it's going to create the charge which allows it to get stuck at the phospholipid bilayer. Now, what is the issue here? The thing is that water is still actually permeable. It's small enough that, once enough charge gets, like if you imagine a phosphate head and you have these water, remember I was talking about solvation shells or ionic shells before. If enough water gets stuck and satisfies the charge difference here at this, it'll get stuck at the hydrogens, right? Because that's where the partial positive charge is. This is negative charge, and positive attracts negative. They'll get stuck like this. That means that there will be places between the phosphate heads where the charge isn't strong enough to attract the water, right? The electric potential, you know, because remember, what is the equation for electric potential, right? 1 over R. So if there's the distance between the phosphate heads is large enough, right? And, you know, water will be able to sneak through, right? Because there are places where water, you know, won't be attracted as much by the potential. Here, it will still be attracted by the potential of the phosphate head, but if there's enough water, right, that water concentration gradient is going to be strong enough to force the water through to counterbalance this electrical potential here that's created by the adjacent phosphate head. So therefore, under normal conditions, you actually do have some amount of water going in and out of the cell. Okay, so that's good. But, you know, again, that diffusion of water, the passive diffusion of water, which counteracts the negative charge of the phosphate heads, is not, and again, water will get stuck here, right, inside, because the phosphate heads also point inside the cell, into the cytosol. But, so water will get stuck, but water needs to get through much more efficiently than the passive diffusion mechanism, okay? In order to do that, there's special channel proteins called aquaporins, which are designed so that water goes single file through the aquaporin into and out of the cell. Okay, these aquaporins are actually quite amazing, right? Because these aquaporins actually, it's interesting because at any given time, they transport in single file. Remember, single file, think about how small that is on the molecular level, like angstrom small, right? Nanometers to angstrom small. Single file, it transports water through 1 million water molecules every second, right? So that's pretty insane, right? Like 1 million new ones of these are going through every second single file. That's astronomically hard to imagine, you know, it's just crazy, right? And this aquaporin is organized in such a way, so this allows basically water to more effectively transmit inside and outside of the cell. So aquaporins allow for both, right? Depending on a lot of different things that are outside the scope of this video. But you might be asking, so when do we need aquaporin activity versus regular good old passive diffusion of water outside or inside of the cell? Well, most of the time, water is going to passively diffuse inside the cell. You need water to be ejected outside, right? So in order to eject water outside, you have a situation like, for example, when you're crying, right? Your body needs to effectively export water from outside of its cells. When you're sweating, right? Water is exported outside of your cells, and through other many ways besides aquaporins, but through aquaporins, right? And in your sweat glands. So basically, there are times when you need to expel water from your body. Saliva, for instance, is another way. And you can't do it, just rely simply on, you can't rely simply on diffusion to do that because it's just too slow. So you need some sort of water pump. All right, so that's enough of that. There's a ton more to discuss here. I'm going to go into sodium, potassium pump, active transport stuff in the next video. We're building to the eternal life stuff. Well, we won't get there for a long time. You have to accept it. That's just the rate at which it's going to happen. And these videos are going to get recorded anyway, but re-recorded, but just accept that this is what it is. See you around.